the two hybrid assay technique. So they had, as we saw again before, how we had when IRS1 and JNK1 came together, we had the transcription of the reporter gene. So this is the exact same thing except with IRS1 and the insulin receptor. So when they come together, we're going to get transcription of the gene and yeast on plate. But this is essentially like a tri-hybrid assay in the sense that we're also adding either active or inactive JNK1 to the mix. So essentially what this top part of the experiment is showing us is that, EV is also a control well, just for all y'all want to know. But so essentially what this top part of the experiment is showing us is that when JNK1 is active and present, it's going to inhibit the binding of IRS1 to the insulin receptor. So now we're moving down here to this well. And so I have that the substitution of serine 312. So again, we're talking about serine 312 is in humans, so it's the same thing as serine 307. So we're talking about 307 here, sorry. So this is in rats. So substitution for serine 307 for alanine in human IRS1 blocked JNK1 mediated growth inhibition. So again, when we don't have this serine 307, we have no problems because the main role is that JNK1 is going to activate, is going to phosphorylate serine 307, which is going to cause all these problems. So when we do alanine mutagenesis on the serine 307, we're not getting any, we're still going to get yeast growth and everything is fine and dandy. Okay. So essentially what this suggests is that serine 307 is required for JNK1 mediated disruption of the interaction. And then we have this last one here, which just says that IRS1, including residues 45 to 516, so this includes serine 312, but lacking the binding homology region of JNK1, so this would be the LXL motifs, had no interruption of interaction as JNK1 could not bind and therefore could not phosphorylate serine 307. So does that make sense to everybody? I know you can't answer me, but hopefully it does. So what this slide is just showing is kind of three separate experiments that they did to show that JNK1 and its phosphorylation of serine 307 in rats or 312 in humans is going to be responsible for the inhibition of the insulin receptor and IRS1 interaction. So I hope that's clear. So again, the first one, sorry if I'm repeating myself way too much, but so the first one we had IRS1 and the insulin receptor, and when we had active JNK, we had inhibition of the interaction. Here we took serine 307 and did a substitution with alanine, and we found that there was no interaction with JNK1, because again we need the serine 307. And here in this IRS1, we just took out the LXL motifs, so again JNK1 was unable to bind, so we had no interaction with JNK1 and IRS1, and therefore the insulin receptor and IRS1 were able to bind, and the whole signaling cascade was able to continue. Okay, so now this one, I think we're almost done. One more slide, guys. So for this one, we're looking at decreased insulin-stimulated tyrosine phosphorylation. So here, this one again, it's a little bit tricky, but we'll get through it. So in this experiment, we looked at something called immunoblotting, which we know hopefully, well, not Adrian, I guess, from anatomy we know some stuff about immunology, but Adrian, I'm sure you'll catch on. You're a smart guy. So in this experiment, immunoblotting techniques were used. So immunoblotting is used to identify target proteins among a number of unrelated proteins. Identification of proteins is going to be by antigen-antibody specific reactions. So anisomycin, which is this guy, anisomycin, was used to activate JNK and promote phosphorylation of serine 307. So essentially whenever you see, so this is going to be the concentration. So here we have, we're going to have very little concentration. So we're going to have JNK1 isn't going to be activated, whereas here we have really high concentrations of this aniso stuff. So we're going to have a lot of J and K1 activation. So hopefully that's clear. So again, right here as well. So here's when aniso is high, that means that J and K1 interaction is going to be high. When aniso concentration is low, that means our J and K activation is going to be low. So, tyrosine phosphorylation of IRS1 was detected by immunoblotting with anti-phosphotyrosine antibodies. Does that make sense? I know you can't hear. So hopefully that makes sense with everybody. So what these strips mean here is this is just showing the results of the immunoblotting. So it'll make a little bit more sense when I explain the experiment a little bit more. So in cells with mutated J and K binding domain, which is this guy here, again, I'm going to have to change the labels or something because this is awful now that I'm looking at it. So in cells with mutated J and K1 binding domain, we see normal tyrosine phosphorylation regardless of the concentration of acino. So essentially what this is saying is that when J and K1 is unable to bind to IRS1, we see very normal amounts of tyrosine phosphorylation. So this is when everything is running super smoothly. Whereas when we go over here, in a non-mutated IRS1, we see far less tyrosine phosphorylation as the concentration of anisomycin increases. This decrease in tyrosine phosphorylation can inhibit downstream pathways, specifically inhibiting P85 from binding to IRS1. So this gets more into our PI3K stuff. So as we can see, the more aniso we're going to add, that means that we're going to have the more J and K1 activation we're going to have, and we see less tyrosine phosphorylation. So again, this is going to be when we have IRS1 with mutated LXL domains, and this is going to be IRS1 with normal XL domains. As we can see, as we increase the amount of aniso, we're going to see a decrease in tyrosine phosphorylation. So again, decrease insulin-stimulated tyrosine phosphorylation. And so these wells right here serve as controls, as we can see, because insulin is either present or not present if they ask. So yeah, and maybe we could take out these labels at the bottom too. Okay, so hopefully that is a good enough of an explanation. As I said before, please comment any questions you have in the comment box, either under this video or under the other one, and I'll try to answer your question as soon as possible. And yeah, so have a good rest of your weekend, everybody.